Hello, my name is Christopher Walsh, and I am a DO PhD student at the Heritage College of Osteopathic Medicine, a part of the medical program and a part of the Translational Biomedical Sciences PhD program. I study the lymphatic system, also known as the sewer of our organs. And the system gets this name because it collects the interstitial fluid, or the fluid that leaks from our blood capillaries into our organ space. It collects all this fluid as it washes over our cells and returns it back into hematogenous circulation. The lymphatic system contains a number of inbuilt pumping mechanisms that allow it to move the fluid from the organ space back into blood circulation. And here you can see a video of the human knee and the lymphatic vessels that course through it, and the pumping mechanism that is moving lymph from the foot back into the heart. The lymphatic system also undergoes growth called lymphangiogenesis, and that occurs when signals from the tissue space make it to the lymphatic vessel and induce growth in the endothelial cells that live there and cause them to sprout and migrate to the area that created the signal. Two of these signals are growth hormone and its downstream partner, insulin-like growth factor 1. Both of the receptors for these hormones have been found on lymphatic vessels and have been found to cause growth in various disease states. However, one thing that I find interesting is that there's not many studies on how growth hormone or IGF-1 affect the lymphatic vessels at normal physiological states. And so I asked the question, how does growth hormone alter lymphatic vessel pumping or wound healing? Thankfully, my laboratory has the perfect mice to study this question. We have transgenic animals that are models of growth hormone overexpression, known as the BGH animal that causes gigantism, models of growth hormone decrease, such as the GHA mouse, and models of no growth hormone action at all, known as the growth hormone receptor knockout mouse. And this mouse is really interesting because not only is the longest lived laboratory mouse, but it is resistant to diabetes and cancer. To look at the lymphatic vessels in these animals, I use near-infrared fluorescent lymphatic imaging. This is a new style of imaging that was created by Dr. Eva Savik-Maraka at the University of Texas Health Science Center, and it involves shining a laser diode at 785 nanometers into the skin, which will fluoresce a tracer that you can inject into the skin that excites a camera, and now you can actually look at lymphatic vessels pump in real time through the skin of living animals. And what I wanted to measure with the near-infrared fluorescent lymphatic imaging is lymphatic pumping rate and lymphatic wound repair in these animals. So here is a video of how to measure lymphatic pumping rate in an animal. There you can see a mouse that was injected with a tracer, shown with the laser diode, and a camera has collected the information sent back. And you can see at different moments there are pulses of bright fluid that pump through that vessel. And you can simply count the number of pulses per minute to get an assessment of the lymphatic pumping rate. And I performed this on each of the animals along with their wild-type counterparts. And then secondly, I used a model of lymphatic wound repair, a simple surgery where you open up the mouse after you anesthetize the animal, you expose the collecting lymphatic vessel, which you can see there connecting points 1B and 2, and you sever the vessel. Next, you close the skin with surgical glue, and you use the near-infrared fluorescent lymphatic imaging to sequentially image the animals as they heal that vessel. And this can give you all kinds of information about how that vessel is able to heal itself. And what I found is that growth hormone action actually correlated to lymphatic vessel growth, and that the animals with increased growth hormone action had increased lymphatic pumping, and that the animals with decreased growth hormone action had decreased lymphatic pumping. And from the surgical experiment, I found that growth hormone disrupted lymphatic wound healing. So this is, again, compared to the transgenic animals versus wild-type counterparts. And I found that the animals with increased growth hormone action and decreased growth hormone action, specifically the GHA animal, had decreased ability to heal their lymphatic vessel wounds and took longer time to heal those wounds. In conclusion, I found that growth hormone, for the first time, alters lymphatic pumping rate and lymphatic vessel wound repair. And lastly, I wonder if this could contribute to the GH-associated diseases that we see in the BGH animals, such as diabetes, cancer, and aging. Thank you.